This protocol describes a method to quantify lipid droplet formation in human intestinal organoids. It can be used as a high-throughput screening platform to test for drugs that modulate lipid droplet formation.
The method we describe is based on a lipid droplet-specific dye and biopsy-derived intestinal organoids. It thereby provides a scalable, accurate, and physiologically relevant model for lipid droplet formation.
This protocol can be used to screen for patient-specific novel drug candidates that modulate lipid droplet formation, for example in DGAT1 deficient patients. This lipid droplet formation assay can also be applied to other types of organoid cultures to study lipid metabolism in other cell types.
The assay is to a significant extent dependent on the density of the organoid culture. Try to ensure a consistent organoid density across samples and experiments. Visual demonstration of this assay is critical because it is important to show how the organoid should be cultured and how they should be handled to ensure proper analysis of lipid droplet formation.
After preparing the organoids, carefully aspirate the culture medium without disturbing the droplets of basement membrane matrix. Add 500 microliters of cold basal medium to the first well of organoids. Using a P1000 pipette, gently pipette up and down to disrupt the basement membrane matrix droplets with organoids. Repeat this procedure with the same medium in the next well as required, but do not harvest more than 2 wells per 500 microliters of basal medium. Collect the organoids in a low-binding 1.5 ml microcentrifuge tube and spin them down in a mini tabletop centrifuge for 15 to 20 seconds.